Our next speaker here is going to talk about a very interesting subject, I think, because it has to do with so much that's going on today in terms of cell health in cancer and neurodegenerative disease. And recently, a very, very good friend of ours had a very serious neurodegenerative disease called Lewy body, for which there was no cure and no hope. So I think the research being done in these fields is so terribly important for more people than we even imagine have some of these afflictions. And when we first met, remember we talked about you're bringing your children to the lab, I think. How many were you were bringing? What, your daughter?
My daughter, yeah.
Your daughter to the lab. And I still think this is a great way of mentoring. I brought my son to the publishing company. He was there for a while now, someplace else. But this is a great way to introduce young people to science, to bring them to get the feel of it. So we're very interested in hearing about how you have managed to combine this and do the advances in the wonderful field that you're in.
So, from what I understand from the instruction that given to me by Marianne, she wants me to tell you how I got here. And I thought about it, and I think it would be a little hard for me to give a purely technical talk to this diverse audience. So instead, I want to go back a little bit further from the origin of my life. And I think because that has a lot of impact on who I am today. So I was born in China, and I spent my first 20 or so years in China. And my first 10 years was basically living in a pretty privileged life because my family were pretty prominent intellectuals in China, and so we were given a living condition that most Chinese were not allowed to have at the time. But my life totally changed when Cultural Revolution started, and I was 7 at the time, and basically both my grandparents were either driven crazy or put into seclusions, isolation for what they had been teaching. And my father was forced to criticize his father, and he under so much stress, and then he was hospitalized and went from a totally healthy 40-year-old to died within 10 days. And one of my grandfathers at the age of 82 was scared so much and went crazy and had to be hospitalized in a mental hospital. So, and that was the start of my elementary education. And my elementary school building also got burned by the student, and so I didn't have a chance to go to my first grade. And so throughout my elementary and middle school education was during the 10 years of a Cultural Revolution. So basically, I really did not learn that much because it was under the slogan of Chairman Mao, the more you learn, the more stupid you become. So we really sort of had a great time and just sort of playing around and not really having a lot of homework to do. So my fortune started when I got into high school. And my high school teacher, Mr. Lu, and for some reason he strongly believed that I should go to college. And you have to understand, it was a very strange notion at the time, and the college completely closed doors during the 10 years of a Cultural Revolution. And eventually, at the end, opened, but the students were all were chosen to go to college, or those have to be from the family of a farmer and workers, and I clearly did not qualify. But Mr. Liu believed so much that I need to go to college, and he plotted various kind of schemes with my mother and trying to get me into college. But unfortunately, because I was the oldest in the family, and according to the policy at that time, I need to go to the countryside, which means that I have to go to a very remote area to become a farmer. And as a student, I really did not understand why I need to go to college, because I really had no notion I can go to college at that time. And actually, it was the cool thing to do is to be a farmer. So I wanted to be a farmer. But my mother and plotted with my Mr. Liu, and somehow they got me into a factory in Shanghai. And so there I started to work on the machines, which I really hated, and I wasn't good at it at all. But I was very lucky, because at that time, Deng Xiaoping got to the power of the country, and he wanted to restart the college and entry exam. Because before that, in the Cultural Revolution, the last few years, they did have college, but the student got chosen to go to college had no exam to pass through. So they just picked the farmers or factories. And 4 months before it was publicly announced that they're going to have the college exam, my teacher got to know this from grapevine. And so he heard this rumor and says, oh, he was very excited to come to me, and he asked me to practice and then doing reviews. But unfortunately, there was just no textbook that I can study, because all the textbook, high school textbook, all burned during the Cultural Revolution. So I had no book, even though my family my grandfather was a writer, and all of his book either taken away or burned. So I literally have no textbook to study. And so my teacher thought of the school library actually had a textbook from before the Cultural Revolution, but the library was sealed, with a big white piece of a paper said, nobody can enter there. So Mr. Liu figured out that somehow he got the key, and he sneaked in there at night, and so he took out, stole all the textbook before the Cultural Revolution and for me. And so I had 4 months to study, and all by myself, because Mr. Liu was a Russian teacher, and so I had 4 months to study, and then before I took the exam, and it turned out that I scored the #1 in Shanghai. So you had to understand that was the context of this exam, because not only the high school graduate of that particular year, 1977, are qualified to take the exam, but also all the students during the 10 years of a Cultural Revolution were allowed to take that exam, because that was the first time. So miraculously, I became the #1 in Shanghai. And when the reporter came to our house to ask me how I did it, so he asked me to show him the notebook I have done during these 4 months. And I took out the notebook, and it turned out like 3 feet of notebook that I did in these 4 months period. But anyway, so I got into college, and 4 years later, and also didn't have any idea that I should come to the West, because it was also not possible. And by the time I about to graduate, so Dr. Manfred Karnofsky and Dr. Ray Wu, so Karnofsky is at Harvard, and Professor Ray Wu is at Cornell, and organized this exam for the biology student. And I was allowed to take the exam, and I scored very top again. So, and that's how I got accepted in the Harvard Medical School in the Department of Neurobiology. Actually, the same department Carla did her graduate study, and later on become the chair. And so in the neurobiology department, and I was first assigned to a professor, but he did not get a tenure at Harvard, so I had to change a lab. And so that's when my opportunity starts. I got to choose what I want to study. So can I have the first slide? So at that time, we had to take a lot of classes, and one of the classes was the mechanism of neurodegeneration. And at that time, I really didn't know anything, about genetics or the mechanism, anything about how these neurodegeneration come about. But I was really struck by the, oops, it does work? No, oh, you clicked. Okay.
Go back?
Yeah, go back, please. So, for example, this normal human brain section, the caudate area of the caudate putamen are completely missing in the Huntington disease patient. So at that time, the Huntington gene was not even cloned at that time, but I was struck by the fact that why the whole brain wasn't completely wiped out by Huntington mutation. Instead, that the only specific area was wiped out. So I became curious that why these neurons selectively were killed by this mutation. And in another class, called the mechanism of neurodevelopment, and taught by Tom Jessel. And at that class, I learned about there are also many neurons die during the development. But again, this is very selective neuronal cell death. But this is a totally different context. This is normal development. But at that time, nobody actually pay attention to how these neurons die. The only idea was to study how to prevent these neuronal cell deaths by adding extra trophic factor, by adding additional targets. So the idea is that these neurons die because they don't have enough trophic factor or they don't have enough target support. But nobody actually pay attention to the death process itself. There was little idea or little notion of why or how these cells may control their own demise. Because people commit suicide. Why would a cell also want to commit suicide? So this is a totally different, the notion that nobody really paid attention to that. So which made me thought about that, this early development, you have very selective cell deaths. And later on in the disease, you can also have selective cell deaths. I began to wonder whether there's any connections. And next. But I look around at Harvard, and nobody was studying cell deaths at the time. And by chance, I heard Bob Horvitz, who was an assistant professor at MIT, was talking about his studies on nematode cell deaths. So I petitioned to our program chair, I was a Harvard student, allow me to work at MIT. So that's where I did my thesis. And by the time of 1990 that I graduated, we appreciate that in the C. elegans, there's a very simple cell death mechanism controlled by sensory and CF4. This was my PhD thesis. And there's also a gene suppresses this cell death paradigm. And in mammalian cells, but the connection to mammalian cells was not shown at all. But it was known that in the mammalian cell, there's an oncogene called BCL2. When you overexpress BCL2, you can prevent cell death. The cells, so the idea was that in the oncogene, in promoting oncogenesis, you can prevent the cells that should have died, then the cells get transferred. But there's absolutely no connection between the cell death pathway in C. elegans and the mammalian system. So that was the first question. After I graduated from Bob Horvitz's lab, I wanted to ask if the C. elegans pathway also exists in mammalian cells, but unfortunately, there's still nobody working on cell death mechanism in the mammalian system. So with Bob's help, I landed my first job right after my PhD, and with the goal of trying to answer whether the C. elegans cell death pathway that I studied as a PhD thesis also exists in mammalian cells. So this was actually my first R21 grants title, to ask whether there is a similar mechanism. And so this, 2 years later, we were able to show that in the C. elegans, the homologue of CEF3 do exist in the mammalian cells. At that time, it's called ICE, and now later on, it's called caspase 1. So both ICE and the caspase CEF3 are cystin proteases, and when cells die through apoptosis, they activate cystin protease. So this was the pathway diagram that we published in the 1996. So these work provide a foundation to understand that, to allow the understanding that the evolutionary conservation of the cell death program from the C. elegans to mammalian cells. It turned out that the caspase in the mammalian cells exactly conserve the function in the C. elegans performed by CEF3 and CEF4, and the BCL2 is a homologue of a CEF9. This was work of another graduate student in Bob Horvitz's lab. But I want to talk a little bit more about a more recent work. Although our initial discovery sort of opened up the whole apoptosis field, there was, when I started my lab, there's probably less than 100 papers on apoptosis, or caspase, there's none. But now it's like 6-digit papers in this area. But what was very disappointing to me, however, even though there is unequivocal evidence that the caspase activation is very detrimental in neurological diseases, nobody was able to develop inhibitors that can be used in a patient to prevent stroke-induced cell death for the purely technical reasons, because somehow because caspase are cystin protease, or the inhibitor of the cystin protease goes straight to the liver, so it doesn't even go into the liver. So we decided that we want to focus on the chemical aspect of the development and trying to ask if we can develop chemicals that can effectively inhibiting cell death, so this that can be developed into the drug one day. So these work represent our effort in this aspect. Our work focused on this TNF-induced cell death. TNF is a very important pro-inflammatory cytokine, is known to involve, for example, rheumatoid arthritis, Crohn's disease, or even Alzheimer's disease. So TNF, stimulation by TNF can lead to multiple downstream pathways. And some of this NF-kappa B pathway is a pro-survival and a pro-inflammatory response. Under certain conditions, cells might undergo apoptosis. So when cells die through apoptosis, you can see very typical morphology of cell condensation. But what's interesting, we discovered that when cells are stimulated by TNF, if they cannot undergo apoptosis by the addition of a caspase inhibitor, you will see inhibition of apoptosis, so cells do not undergo apoptosis anymore. But in a lot of situations, the cell will still die, but with a distinct morphology that we could call it necrosis. Because apoptosis is cell condensed, necrosis is cell lyse. So it's almost the opposite of apoptosis. So what's interesting is that apoptosis has been well accepted as a regulated form of cell death, but necrosis has traditionally believed to be a form of a passive cell death. Therefore, it's not regulated. But the paradoxical finding is actually necrosis is also very common in human diseases. You know, the acute pathologic injuries of stroke, heart attack, there are a lot of necrosis, but also in cancer cells, it's well established that the solid tumors with a lot of necrosis are indication for the bad prognosis. So, but because this notion that the necrosis is just a form of passive cell death, actually nobody paying attention to this form of cell death, So, cut the long story short, we developed inhibitors that inhibit this form of necrosis that we call necrosnamin-1, and this is very popular chemicals in the cell death field. It turned out that the NEC1 is very strongly inhibiting necrosis. So for example, this is the paradigm of necrosis. You can see cell lyse, and when we add NEC1, it doesn't, cell looks completely normal. When the apoptotic cells, if you add NEC1, it doesn't touch it. And these are not only morphological protection, but the cells actually in the present NEC1, it can proliferate when without NEC1 will die completely. So these are the so far all the published example that people use NEC1 and show that the protection in the paradigm of a cell death. So now it's well accepted that this necrosis is actually yeah, more click, is a regulated form of necrosis that we call necroptosis, and this can be inhibited by NEC1, which turned out to be an inhibitor of RIP1 kinase. The amazing thing is that through medicinal chemistry work, we were able to improve NEC1. Now it's very selective, low toxic, and binds very tightly to RIP1 kinase with KD of 3.1 nanomolar. And some time ago, we did a profiling with this California ambit who has all the kinases in a human genome, and that one turned out to be the most specific kinase inhibitor that human ever developed. So now it's well accepted that in the downstream of the TNF pathway, there are two cell death pathways. One is called the apoptosis, mediated by caspase, and the other one is called the necroptosis, mediated by RIP1 and RIP3 kinase. So we have a movie and shows that this is TNF receptor, TNF bound to the TNF1 receptor, and it leads to recruitment of intracellular signal complex, including TRAD, TRAC2, CIP1, and RIP1, and CIP1 ubiquinates RIP1 kinase, and then this RIP1 leaves the complex 1 to interact with RIP3. RIP1 is deubiquinated and interact with RIP3, and this complex in turn to lead to necrotic cell death. So it's all kinds of phosphorylation events that we still don't quite understand, and cell die by necrosis. Okay, next, yeah. So what is the physiological function of a necroptosis? Next one. And the consensus is that the apoptosis is a mechanism to get rid of a viral infected cells, but lots of virus has apoptosis inhibitor. So when cells infected by virus, they cannot die through apoptosis. So necroptosis may be a backup cell death mechanism, and because necrotic cell deaths are better activated than dendritic cells, so it may act as a better alarm system to alarm our body for the infection of a virus, where the apoptosis is immune silent. Next. And this is our initial work, and then showing the necroptosis blocks the ischemia damage using this mouse model of middle cerebral artery occlusion. And we can show that 6 hours after onset of the injury, we can still protect by NEC1. And this work is now being extended by multiple labs to different models of ischemia injury. And the most exciting result right now, this is a work that we are doing a revision for Nature, is that we found the new finding is a RIP1 kinase mediates oligodendrocyte cell death. As you know, that the oligodendrocyte cell death is a major problem in multiple sclerosis. The #1 neurodegenerative diseases affects young people in this country. And all of the MS drugs so far impacts the immune system because we don't know how oligodendrocyte dies. But it turned out that NEC1 has effect in multiple models, animal models of multiple sclerosis. And so I want to end here. Next, I want to acknowledge many students and postdocs and my collaborator for contribution to this work. Thank you.
So do you see any use for this in cancer and how necrosis?
Yeah, so the problem with cancer is that there are clearly necrosis in cancer, but there's no good model for necrosis in cancer. Necrosis in cancer has been suggested to play a role in metastasis. And there are people, I talked a couple weeks ago, I visited Cleveland Clinic, and one of the senior faculty there is developing a cancer model that involving necrosis. So it was interesting to test it.
And that way you probably want to enhance.
No, you want to inhibit necrosis because necrosis leads to inflammation, and which stimulates cancer growth.
Sort of a balance.
Yeah.
Inflammation necrosis is both involved in cancerogenesis as well as in like cancer development. Because you mentioned that it's usually metastatic cancer, right?
Yeah, yeah.
Already,
Yeah.
like
Yeah.
the inflammation process is already.
The role of necrosis in the cancer development is not really clear. So one question or one observation is that immune cells, myeloid lineage cells, are very sensitive to this pathway, but all of the cancer cell lines, which are epithelial cancer cells, do not have this pathway. So, but the primary epithelial cells do have this pathway. So, one possibility is that the inhibition of necrosis is actually a requirement for the epithelial transformation or establishment of cancer.
So how conserved is the necrotic pathway? Is it C. elegans in it?
The C. elegans does not have death receptor mediated cell death pathway, so it doesn't have TNF, it doesn't have virus. The C. elegans also have additional cell death pathway that's involving expression of cell death involving LPD1, but not fully. And there's also linker cell death, which involves polyglutamine kinase molecules. But how exactly that is conserved in the human is not clear. But in Drosophila, there are TNF, but we cannot find the exact molecule like with one. But we can sort of find the kinase activity and then the death domain maybe on the two different molecules. So it's possible that in blood, this component is separate, executed by two molecules.
Have you looked at the process of cell death in ALS at all?
Yeah, that is a very interesting question that's ongoing. We're building a model. I think it is.
Okay. The existing models. Really
Yeah, we're building a model.
I have to ask what happened to Mr. Lin, but also I want to know if you think you're going to go back to looking at Huntington's also.
So there is a paper by former postdoc of Robert Freedom is now, and it says that network loss of these others. So, but we have
What about Mr. Lin? I mean, Mr. Liu.
I visit him. He retired. I also go visit.
So he never got in trouble
He's
for
a-
No.
Did your grandfather teach at Fudan?
Yeah. Yes. Yeah. He was actually the county chair of the Chinese university.
So was it closed down entirely during the Cold War?
Yeah, for about 7 or 8 years, there's no student whatsoever. But at the end,
books, and they getting farmers' son, workers' son to go to college to do really study very vigorously. So only after 1977 when Junying had to come,
He was mayor of Shanghai. Where was he?
He was a journalist. He's army. He's really. But
Who was
he
the-
studied in